Great. Good afternoon, everyone. The after-lunch slump is always real. So to make it a little bit more interesting, in a TB conference, we're going to be discussing sperm and sperm counts and everything that goes along with that. So hopefully that wakes you up a little bit. And essentially, why do we care about sperm in TB? I'm not an endocrinologist. How did I get into this? Well, it turns out that Pretomanid had a safety signal in rodents, and that's what we're coming to discuss here today. All right, so essentially, we know that BPaL or BPaM regimens were promoted and said to be programmatically implemented by the WHO in May 2022. By a show of hands, who has prescribed BPaL at least once for a patient? Okay, clinicians in the room, thank you for the hands, great. Okay, BPaL, it works like a bomb, right? Is it available to everyone? No, we do not. It is not currently indicated for patients under 14 and pregnant women. Why? Because there was the signal of testicular toxicity in rats. So, very, very high level, I want to leave you with a couple of quick impressions. First of all, Pretomanid is a novel nitramidazole, and it actually looks like this might be a class effect. Another drug is metronidazole, which also had testicular toxicity affecting the seminiferous tubules in rats. And did it actually ever pan out in humans? No, it didn't. Do we prescribe flagyl metronidazole to children? Absolutely, we do, yes. Okay. So importantly, the changes were seen in seminiferous tubules. And for those who haven't done anatomy in many, many years, that's basically like the lining of the tubules inside the testes. Okay, and what we see is that the germ cell is degenerated, which affects spermatocytes and spermatids with decreased fertility, but it was reversible. And so we also saw from the rodents, rats and mice, that it was dose-related. So it only happened at very, very high doses. At the bottom of the slides, for those who do not routinely work with pharmacokinetics and pharmacodynamics, there's this thing called the NOAEL, all right? So that is the no observed adverse effect level. Essentially what that means is at this level, below that level, there were none of these side effects. And that level in rats was 80 megs per kg per hour per mil. The target that we aim for in humans is 50, so substantially less than where we were seeing the side effects in rats. And so the short version of this is that the exposures attained with Pretomanid in humans are far lower than drug levels in rodents where no effects were seen. Okay, so that's a little bit of the preclinical background, so the background in non-humans. This looks very confusing, and essentially we just need to remember that there are negative feedback loops and positive feedback loops when it comes to male anatomy. Essentially, things start happening in the brain, which affects the testes, but if you were to see any damage to seminiferous tubules in men, you would expect inhibin B to go down and therefore FSH to increase. That's the short version. Okay, so those are good markers of seeing if there's any toxicity to be seen in testes. And what did we see? So in preceding trials where Pretomanid was given in Pretomanid-containing regimens compared to standard of care, where they used Rifafort, essentially, did we see the INHA going, the inhibin B going up? No, we did not. Going down. Did we see the FSH going up? No, we did not. So essentially, what we did see when we did these hormones in men taking Pretomanid-containing regimens is that before treatment, many of these patients were borderline hypogonadal with testosterone levels near low limits of normal. In addition, we also did some paternity surveys, and essentially, and everyone in clinical trials here knows, despite doing our utmost best to tell people not to have babies, they went ahead and had babies. Okay, so there were 45 children fathered by men who had taken Pretomanid, okay, against our best intentions, which we know happens. Okay. Just to note that this was not during the clinical trials, this was a period after. Okay, these were not multiple pregnancy reports that stressed out all the investigators. So the results for this trial was it was a standalone study looking at testicular safety. And why was this done? Because when we wanted to start looking at using Pretomanid in children, the FDA felt that the data just weren't strong enough. We needed to actually get sperm samples from men with drug-resistant TB actually receiving Pretomanid. So with as much enthusiasm as we could, there we went. Essentially, the primary endpoint was the change in baseline from total sperm count to week 26, which was the end of treatment. Secondary endpoints looked at the same thing at different time points, so sperm count change from baseline, but also concentration, which ended up being a lot more useful. Interestingly, for study design, why did we not just use this in BPaL? Well, it turns out that linezolid actually has a toxicity signal for that same seminiferous tubal toxicity in preclinical models, not that anyone seems to be too concerned about that. So we used it in BPaMZ. And then, interestingly, just for your information, why were we intent on doing those semen samples at week 44? Well, because it represents a potential recovery of semen parameters. So it's 1.5 cycles of seminiferous epithelium, so basically the cells are turning over and renewing after we stop the Pretomanid, and it also includes the epididymal transit time, so the time from sperm creation to be able to get out. In other words, if Pretomanid was causing a big problem, we would have actually seen a recovery after stopping the Pretomanid. 26 men were enrolled at 4 different sites. I know that these are low numbers, but it was actually a very difficult trial to do. And it was also very difficult to statistically plan because we didn't know what normal values were, and I'll go into that in a little bit more detail. Essentially, if you had RIF mono, INH mono, or MDR-TB, you could be enrolled to the trial because it was a fluoroquinolone backbone trial. The different crosses on the slide show at which weeks we did the samples, and so the men were asked to do 2 samples, at least 48 hours apart, with abstinence in between, and the mean of the 2 was taken as that week's sample estimate. The numbers were small, as I said, and there were a high number of screen failures for multiple reasons, most of which were azoospermia at baseline. It turns out that men who are unwell with drug-resistant TB are not great at producing samples with high sperm counts in them. As we know, when you get better from TB, you gain weight, women get their periods back, your hair gets shiny, and your sperm count goes up. Go figure. So the challenges were substantial. It was complex to implement just because of lack of experience in this. There was a large variability in sperm count. We know it goes down with age. We know there's variability between people. And it was also difficult to determine futility results up front. Due to time, I'm just going to skip a little bit over some of these slides. And essentially, the baseline demographics were typical of a drug-resistant TB population. The primary endpoint basically showed us that change from baseline, sperm count went up to week 26. And here you can see a shift in the sperm count distribution, showing that the average was also increasing. And this plot is just an interesting plot because it shows us that the sperm count plotted against the actual amount of Pretomanid in the blood, there was no correlation with that whatsoever. And the secondary endpoint, which are the final results, which we're really focused on today, is that there was no massive increase between week 26 and week 44, showing that Pretomanid was causing some kind of damage, which was encouraging. And so really what the study did is that it didn't show statistical significance because that would require thousands of people because of the innate variability in sperm count, but it did show us a trend. And it showed us that the adult males with drug-resistant TB treated with the BPaMZ regimen for 26 weeks had improved spermatogenesis with a mean sperm count increase. They had massive variability both within and between participants. There was improved gonadal hormone status and BMI, so all the hormones did the right things and BMI increased. Notably, all participants culture converted. We know that the BPaMZ regimen is an absolute bomb, but we need to be careful with liver. But there were no new safety signals in this regimen that we hadn't seen before. So in conclusion, this was the first time I think anyone's ever tried to take sperm samples from men with drug-resistant TB. We didn't know what normal was, and we tried to figure it out as we went along. But I think that these results show from just a physiological basis, but also from a result basis, that probably we're going in the right direction. I think due diligence has been done, and Pretomanid pediatric studies need to get going because BPaL needs to be available to every type of person, including the kids. And I would just want to thank sponsors, implementing partners, sites, participants always, and their families. Thank you.